This video is about proteins, and what we're going to specifically talk about with regard to proteins is not only their structure, but a quality called marginal stability. And we mentioned this when we talked about the functions of maintaining homeostasis, right? We had the qualities of the internal environment being regulated, and one of the key purposes or adaptive purposes of that regulation is to maintain protein function, and that boils down to the characteristic called marginal stability. Now, they have proteins have a broad importance. Let's talk about that first, which means that they're involved in a variety of functions, one of which is structural. There are proteins that serve structural functions. Cytoskeleton would be a very good example. We have proteins that serve functional roles. And there we have a whole array of variety. We could think about things like enzymes, channels, pumps. We can go on and on. Transcription factors, a whole variety. And then really the last category that I would point out would be regulatory. There are proteins that serve regulatory functions. And sometimes they're dual. In other words, there are structural molecules that have enzymatic function, for instance. So again, there's some crossover here, but those are the key important functions of proteins. Now, proteins are the only organic molecule coded for by DNA, right? So we know that we have DNA. DNA is used to make mRNA, right? Only of the DNA transcript components that we want to convert into protein, right? Not all DNA is transcribed in any cell. And so we have this select group of genes that are transcribed to mRNA, and then of course those are translated into protein. And when we do this, we start out by making when we make proteins, we start out by making a sequence of linked bonded amino acids, right? And so, for instance, right, we could draw sort of a structure here that has bonds, meaning the lines, and then the balls, meaning the amino acids. Now, I can't go on and on with this, but if we're going to talk about proteins, we would be talking about linkages of at least 50 or more amino acids. Anything smaller than that, and it's called a peptide. There's no real difference besides the size between a peptide and a protein, right? So this would obviously be a peptide because it only has about 8 or 10 amino acids there, right? But that would be called, and again, let's put dot, 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 right? This will continue a longer sequence, but this is referred to as the primary structure, right? So functional proteins do not only have primary structure, they have to have structure developed beyond that, which means we have to move into secondary structure. And that's going to include two main characteristics. They're called alpha helices and beta sheets. And really, so they are kind of what they sound like, right? So for instance, the alpha helix is a spiral, and the beta sheets are these sort of wavy patterns where the sequence sort of folds back over on itself, right? And it can continue in that direction, off in this direction. Same thing with this other one. Right, this could keep going. we have this, and actually we could link these, right, in the sequence of an individual protein where you have alpha helix in one region, and then sort of this spanning section, and then a beta sheet, and then maybe another alpha helix. And so that's secondary structure. And still, those are not functional proteins. They've just sort of started to fold up. The tertiary structure comes when we fold the secondary structure. And you can even think of this as already being, right, so in other words, that these individual sections, right, that is secondary structure. The fact that this is sort of folding up with a beta sheet would indicate that, right, we're getting tertiary structure forming here. In other words, of the whole thing, then we would have some tertiary structure, right, because it's folding up on itself. And then lastly, we have what's called, potentially, we have quaternary structure. And that's when you have two or more polypeptides. Well, it's a polypeptide. Polypeptide is just another name for a protein. And so when we have two or more sequences, more than 50 amino acid sequences, that become linked together, we have what's called quaternary structure, or what's called a multimeric protein. Right? And sometimes people refer to these as proteins with subunits. Right? Hemoglobin is a really good example of a protein that has multiple subunits. It has quaternary structure, and because it has 4 subunits that are linked together. Now, okay, so we've got this sort of basic idea here behind protein structure, right? We have to get to tertiary structure to have a functional protein, and then sometimes proteins will have quaternary structure if they're a sort of mature form, is when multiple polypeptides become linked. But one of the key things about protein function is that proteins often, most of them, need to change shape as they function. Some of the structural ones don't need to, but a lot of the functional ones tend to change shape in the process of doing their job, doing their function. For instance, channels open and close. Well, to open and close, that's a structural shift, right? A change in what's called their conformation, their shape, right? Conformation. Equals the shape of the protein, right? And so these proteins need to change shape. Many of them need to change their conformation or shape in order to function. Enzymes are a good example. They have a binding site for a substrate that are going to modify, right? And if you've heard of it, there's a model called induced fit, where the substrate binds to the protein, the enzyme, and in the process of doing so changes the shape of the protein, the enzyme. And then when it cleaves it, let's say the job of the enzyme is to cut that substrate into 2 or more parts, it's going to change shape again, right? So the point here is that proteins cannot be rigid structures. They need to have some flexibility. They need to be able to change shape. Well, what generates this shape? Well, the key here, something I haven't pointed out yet, let me put a color on this, right, is that these regions, right, so the blue line here is the sequence of amino acids. We could put, if you'd like, we can put amino acids, little balls on the line here to indicate our individual amino acids, right, linked. Now, the thing is, for it to fold into this particular shape, right, the side groups of the amino acids have to become linked. And that happens through what are called weak bonds. Right? And there's a variety of weak bonds structures. Right? That is a weak bond. And so there are hydrogen bonds, there are what are called van der Waals forces, there are hydrophobic interactions. What that means is that the amino acids that don't like water, hydrophobic, they're water fearing, will sort of like glom together, right? They'll form these weak bonds that hold the protein in particular shapes, right? And so weak bonds are absolutely essential to this secondary, tertiary, and even potentially quaternary structure. Now, the thing with weak bonds is they're weak, right? And so we have this potential to disrupt them. Aha, now we're getting to this, this idea of marginal stability. Let me clear some space here. Right? And marginally stable, marginal stability, means that they're just on the sort of positive side of not falling apart, right? And that's essential for many proteins, not all, but for many proteins, because again, they can't be so rigid that they can't change shape. They can't have weak bonds being altered so they can change shape. Okay, and so that's absolutely essential. And the structure of the protein is essential, and the fact that we maybe need to modify that shape in order to have the protein have its function. And so we have this balancing act in the forces that hold a protein together and the forces that are tending to make it fall apart, right? And so forces in protein structure. Right? Forces that are affecting protein structure, right? And so one would be the weak bonds. And the thing here is there are many, or I drew one here, right? We could draw many different weak bonds here. They could be forming here, here, all over the place, right? Lots of them. So a big protein could have hundreds and hundreds of weak bonds. And you would think, okay, well, they're each adding to the strength of the protein, sort of holding it in its particular shape, right? Which would make it more and more rigid. That's true. Why, right? And so the weak bonds do hold it in its particular shape, but we can't have it be so rigid. So what's the counterbalancing force, right? So these would strengthen the conformation, right? The other force tends to push things in the opposite direction, and it's called conformational entropy. Okay, so we know what the conformation is. That's the shape. Entropy is a thermodynamics term, and what it means is a tendency towards disorder. A really good example is if you clean up your house or your apartment, clean your kitchen, it's nice and neat. Does it stay that way? Of course not, right? Because there's energy input in a variety of forms. You know, your roommates, the rest of your family, your cat, your dog, right, are all tending to make it become less ordered, creating a mess, right? And so entropy is this tendency towards disorder. And so what this means is conformational entropy means that the more ordered a protein becomes, like this example we have here, right? This thing is fairly ordered. It's got an alpha helix. It folds in a certain way up against. We could put a weak bond between here if we wanted to, right? Weak bonds crossing over, right? But it's folded in a particular shape and it's held there by the weak bonds. But the more organized it becomes, the more it's likely to become disordered again, right? It's kind of I like to use the analogy of a house of cards, right? You build a house out of a deck of cards, right? And when it's got 4 cards in it, okay, it's unstable, but there's really only, right, a few cards that could fall over. Now build that structure higher and higher and higher, right? It becomes more and more ordered, which means it becomes more and more unstable. Its conformational entropy is changing as it becomes more ordered, and that tends to weaken the overall structure. Right? It tends to make the house of cards fall apart. And so the weak bonds are opposing this conformational entropy, opposing an increase in its entropy, right? These tend to make the house of cards hold together. These tend to make the house of cards fall down. The good news is the weak bonds are winning, right? Otherwise, your proteins would denature, right? You've heard that term before. That's when they sort of fall apart. And when they fall apart, they can't function. Okay, so let's bring this full circle now, right? What's this all so important for, right? Yeah, okay, we have this balancing act between the weak bonds and a conformational entropy, making it marginally stable, right? Because the weak bonds are winning by just a little bit. But it's really important because, let's see, if we have a change in pH, if pH were to fall, well, pH is a measure of H plus concentration, right? So brackets here mean concentration, right? It's a concentration of free protons. Well, if we become more acidic, right, a decrease in pH, the H plus concentration goes up, right? If that goes up, think about this. What did I say? These weak bonds, many of them are what are called electrostatic, meaning it's an attraction between positive and negative charges. Check it out. Positive charge, guess what? It is going to compete with the components here that are forming this particular weak bond, or any of these particular weak bonds. So as we change this concentration, we tend to disrupt the weak bonds. If we disrupt the weak bonds, right, we're weakening, right, because these add strength, right, and then all of a sudden our entropic forces are going to win and the protein could fall apart. This is why we regulate pH. This is why we regulate temperature. This is why we regulate a lot of waste levels, right? Certain electrolytes are very destabilizing. Again, electrolytes, they're ions, they're charged, right? And so because they're charged, they have, right, sodium has a charge. Chloride has a charge, right? These have the potential to interact with these weak bonds and disrupt them. And some ions are more disruptive than others. And actually, some ions tend to strengthen protein structure. And we won't get into the rationale there, but there are differences in what the microenvironment components do to the protein structure. But in the end, the whole point is that we are regulating the microenvironment through homeostatic regulatory mechanisms to maintain levels of ions and pH and temperature that are not incompatible with the protein structure and function, right? Now, last thing I'll say here is that there are occasionally, and not, I shouldn't even say occasionally, there are many cases where there are intentional alterations of the microenvironment or even alterations that are adaptively utilized, if you will, by the proteins to adjust their structure and then adjust their function. Hemoglobin, we're going to talk about this when we get to hemoglobin. Hemoglobin, right, so the local environment produces, right, carbon dioxide, right? Your cells produce carbon dioxide, right? Hemoglobin, HB, abbreviation for hemoglobin, carries oxygen, right? And the more oxygen the more readily it binds oxygen, the better in terms of delivering it to the tissue. But then once it gets to the tissue, we want it to unload this oxygen, right? We want this oxygen to come off, right? At the tissue, we want this guy to disengage. Guess what? If carbon dioxide binds to hemoglobin, it makes this breaking of this connection here more easily removed, right? It's called a decrease in affinity. This oxygen comes off more readily. Okay, so again, sometimes we use changes in the microenvironment, CO2, H+, any of the others, to actually intentionally, quote, evolutionarily intentionally, modify protein structure to give us a beneficial function, right? O2 unloading at the tissues, right? Over here to a cell. Okay.